Good morning, everybody, and thanks for waking up early to come hear this session. And special thanks to Norberto for inviting me. It's been a real pleasure to be here and get to see your university and just kind of get to be immersed in the culture here. Today I'm going to be talking about molecular biomarkers of SUDEP, which, as she just mentioned, is sudden unexpected death in epilepsy. As it pertains to the brain and the heart. And so just to give you an outline of what I'm going to be talking about, I'll first define what SUDEP is, and some of you may not be familiar with that, and I'll discuss what the incidence of SUDEP is, how often it occurs, and then what some of the mechanisms are for SUDEP, or what we think the mechanisms are. And then I'll introduce this idea of the biomarker, specifically genomic biomarkers, and then I will discuss some of the genes that could be potential candidates for genomic biomarkers in SUDEP, and then I'll select two that I have personal experience working with to go into more depth and discuss what we know about them. So to begin, let me define what SUDEP is. So as I said, SUDEP stands for sudden unexpected death in epilepsy, and let me see where my pointer is. So right here is kind of the textbook definition of SUDEP, but in more layman terms, any death in somebody with epilepsy who is otherwise healthy that is unexpected and unexplainable by any sort of anatomical or toxicological explanation is classified as SUDEP. And so a typical SUDEP death may go something like this, where somebody with epilepsy who is a healthy individual other than their epilepsy, maybe they go to sleep at night, and then in the morning, a roommate or a parent realizes that they haven't seen them, and they go to check on them, and they find that they have passed away in their sleep, oftentimes with some sort of evidence of a seizure. And so, as I was saying, SUDEP, a big component is the fact that when you go and do an autopsy on people who are suspected to have died of SUDEP, you cannot find any sort of explanation in the postmortem examination for why they died. And so some of the risk factors that people have come up with for SUDEP are people of younger age, usually between the ages of 20 to 45. It usually occurs in people who tend to have generalized tonic-clonic seizures, whose seizures begin early in life, early onset. As I mentioned, SUDEP usually tends to happen in people in bed, probably while they are sleeping. Other risk factors include drug polytherapy, meaning you're on multiple drugs, and this can kind of be an indicator of a severe seizure disorder. And then also subtherapeutic drug levels, which is kind of an indication of poor compliance with the drug regimens. And so all of this taken together, the general consensus is that SUDEP is somehow related to seizures and a seizure-associated event. And so how often does SUDEP occur? Well, in general, people with epilepsy have shortened lifespans, and that's kind of a fact that usually is not talked about or acknowledged. And so if you look at people with epilepsy, in general, there's about a threefold greater risk of dying prematurely compared to the general population. And then if you subdivide this into people with epilepsy who die suddenly for unknown reasons, the incidence can go up as high as 24-fold compared to the general population. And so if you break that down into numbers, about 1 in 500 people with epilepsy will die from SUDEP every year. And then if you look at the particularly high-risk groups, which include people whose epilepsy is very poorly controlled, the incidence can be as high as 1 in 100. And so all of this makes SUDEP actually the leading cause of epilepsy-related mortality. And so while I wouldn't say that SUDEP is common per se, it is not uncommon either. And unfortunately, it's not usually communicated to patients that this risk exists, because in general, clinicians, we don't know how to predict who is at risk, and we don't know how to prevent it. And so it's usually not talked about. And so one of the mysteries of SUDEP is that in essence, it's kind of a black box phenomenon, meaning we don't really understand why it occurs. We have the main hypothesis is that somehow the seizures are causing abnormal respiration or a cardiac function that leads to death. So the respiratory system and the heart are the two main organs that are looked at with relation to SUDEP. And so for my talk today, I'm not going to focus on respiration. Instead, I'm going to focus on what I would call the neurocardiac axis, which is this brain-heart axis. And so that's not to say that respiration is not important, because it most certainly is, but just for simplicity, I've decided just to focus on the brain-heart connection. And so as you can see, the brain and heart are connected via the autonomic nervous system, comprised of the sympathetic and parasympathetic branches. And so when you go and look at, when you record EEG and ECG in patients, it's actually a fairly common finding to see ECG abnormalities during seizures. Up here is a trace from a human patient during a seizure, and as you can see in this bottom trace, this is the ECG showing their heart beating. And then somewhere about here, their heart stops beating for a period of maybe 20 seconds. And so this is just kind of an example of some of the types of abnormalities you can see. And so this breaks down to about 20% of patients will have some sort of bradycardia or asystole during seizures. There's also been a recent paper that showed that epilepsy patients actually have a threefold increased risk of sudden cardiac arrest, which also highlights this kind of brain-heart interaction. And then importantly, there's been one case report of a kind of a rare witnessed SUDEP event in which a patient had a seizure that provoked an asystole and then proved to be fatal. And so as I talk about this brain-heart connection, I wanted to just kind of highlight some of the complexity involved here. It's easy to just say we're talking about brain and heart, but when you really get into what that means, there's a lot of different sites of dysfunction that we could consider. And so in my research, I like to subdivide things into the brain and the heart and then the nerve connections. But as we talk about brain, obviously, there's many different regions of the brain that could be involved. You could have the cardiac preganglionic neurons in the brainstem, such as the nucleus ambiguus or the vagal motor nuclei. You also have to consider regions important for sensory integration, such as the solitary tract. You have limbic regions, such as we've discussed here, that can ultimately have outputs to the heart. When you consider the nerve, you have to consider whether you're talking about parasympathetic or sympathetic effects. You also could have dysfunction at the level of the axons themselves, the way action potentials are conducted, or you could have dysfunction at the level of the synaptic terminals. You'd also have to consider whether you're talking about motor or sensory axons. At the level of the heart, you obviously have myocytes, but you also have the pacemaking conduction regions that could be affected. And then there's also the little understood intrinsic cardiac neurons, which could also play some role. And so, as I was mentioning, we don't really understand SUDEP well, and we definitely cannot predict who is at risk very accurately. However, there's been a push to try to identify some sort of biomarkers that we could use in patients to try to more accurately predict who's at risk, so that we could intervene to try to keep them from dying from SUDEP. And so let me quickly define what a biomarker is. This is, again, one of the textbook definitions, which is a characteristic that's objectively measured and evaluated as an indicator of a normal biological process, pathogenic processes, or pharmacologic responses to a therapeutic intervention. And so in SUDEP, there's been a number of physiological biomarkers that people have tried to measure to see if they would correlate with an increased prediction of risk in SUDEP. Some of these things include looking at heart rate variability, which can be a measure of sympathovagal tone. And so people have looked at interictal heart rate variability, or this other more complicated variable called the RMSSD, which stands for the root mean square of successive differences in RR intervals. So basically they're just looking at the difference in the timing between heartbeats. All of these are indications of how well the autonomic nervous system is working. People have also looked at the postictal period as trying to be a predictor of SUDEP. That has showed some promise. People have measured heart mass in SUDEP patients, which they can't find any differences. Looking at hypoxemia in the brain tissue has been something people have looked at. There's also been people have looked at serum magnesium and nocturnal seizures. And so all of these things may have some ability to help us understand SUDEP and who's at risk, but none of them have been extremely reliable in accurately assessing that. And so another type of biomarker I'd like to introduce is this idea of a genomic biomarker. And so a genomic biomarker, the definition is that it's a subtype of biomarker that involves a measurable DNA and or RNA characteristic, such as a single nucleotide polymorphism, nucleotide deletion or insertion, copy number variations, or cytogenetic rearrangements. So essentially we're talking about gene variants or gene mutations that could act as a biomarker of SUDEP. And so I'll be upfront and say we're not close to finding these yet, but given the way that we're now able to genotype people much more quickly and cost effectively, the potential existed in the future if we could find a reliable genetic variant that could act as a biomarker, it could be very useful. And so some of the benefits, if we can find these types of biomarkers, would be that we could have more of a personalized way to diagnose patients and treat them. It would give us a better idea of who is at risk. It could give us information as to drug interactions if there's a particular drug that maybe we should or shouldn't give a patient based on their genetics. And also it might help patients, because if you remember, one of the risk factors was people who don't adhere to their drug regimens. And so if they knew they had some sort of genetic variant that put them at high risk, maybe they would be more apt to stay on their drugs like they should be. And then finally, it may help us be able to intervene early on so that we could perhaps implant some sort of device in the patients who are at risk, or give them some sort of treatment that could prevent them from ever getting to the point of SUDEP. And so in trying to think about genes that could be genomic biomarkers for SUDEP, I've done a literature search to try to look for genes that are co-expressed in brain and heart, or affect brain and heart, that have been somehow linked with sudden death and epilepsy. And so in doing a literature search, you come up with about nine genes that have been linked to SUDEP with some degree of expression in brain and heart. And so this is a list of those genes. And I realize this is probably the first time the word gene has been mentioned in this conference. And so I'm happy to introduce the concept, because I like to think about things as from a genetics point of view. And so as you look at this list, I've kind of subdivided it. It becomes clear as you look at these sets of genes that some of them are predominantly brain genes. And by saying that, I don't mean that they're not expressed in heart, but they have much higher expression levels in brain, and we understand their role in the brain much better than we do in the heart. And then conversely, the heart genes show very high expression in heart, and we understand their role in the heart well, but then in the brain they're expressed, but we don't understand really what they're doing there as much. And so I'd also, as you can see in the table, I've highlighted the different sorts of phenotypes that have been shown for each of these genes and whether they've been investigated in human or mouse. And so I've shown it by their expression level in brain and heart, as well as whether epilepsy has been seen in humans or mice, and then whether there's been a cardiac arrhythmia documented in humans or mice, and then finally, whether their link to SUDEP comes from human or mouse models. And so I'd like to point out too that of all of these genes, 8 of the 9 encode ion channels. The only one that doesn't is this more newer gene that's been identified called PRRT2, which is thought to be a presynaptic protein. And when you look at the heart genes, another interesting thing is that 3 of the 4 are known to cause a cardiac disease called long QT syndrome, which we'll talk about more later. And so for the rest of the talk, I'm just going to focus in on one gene in the brain class and then one in the heart class. And these are two genes that I've picked because these are ones I have personal experience working with. So the first gene that we'll discuss is KCNA1. And so this is a gene I've worked with a lot in my lab, and this gene encodes Kv1.1 voltage-gated potassium channels. These channels tend to form heterotetramers with other Kv1 subunits, such as Kv1.2 or Kv1.4. And as I mentioned, I call it a brain gene because prior to some of the work we did, it was considered primarily to only be expressed in the brain with little to no expression in the heart. However, subsequently, we have detected it in mouse heart, and we still don't understand what it's doing there, but we know that it is there. In the brain, you can see the genes widely expressed, especially in limbic regions. It's especially localized along axons. In myelinated axons, it's located underneath the myelin at juxtaparanodes. In general, this gene functions to dampen excitability by regulating action potentials, repetitive firing properties, and transmitter release at terminals. And so epilepsy has actually been seen related to this gene in both mouse and human models. It was first actually identified in mouse in a knockout from 1998 where they specifically knocked the gene out of mouse and found that it resulted in very severe spontaneous generalized tonic-clonic seizures in these animals occurring about once every 2 hours. And so that's an image there showing you of kind of what one of these seizures typically looks like. And then I'd like to point out that this is kind of an example of a bench to bedside where the gene was found to cause epilepsy in the mouse first and then subsequently found to cause epilepsy in humans based on that mouse work. And so then there's been at least 3 families that have been found in humans that have complex partial epilepsy due to disruption of the KCNA1 gene. So this is a human epilepsy gene. And so I wanted to show this. I'm sure all of you have seen mice having seizures, but I just wanted to show this recording of one of our mice that lack KCNA1 having a seizure. Let me go back here. And the reason I wanted to show this is because I know there's a lot of work being done in pilocarpine and inducible models here, and I just wanted to show this to emphasize the fact that these animals, these seizures are not induced. They lack a particular gene, KCNA1, and it causes them to have these seizures spontaneously without any sort of provocation. And so this is showing some of the convulsive episode during this generalized seizure. And then now the seizure ends and you get a period of EEG suppression. And so the reason I got interested in looking at these mice in the context of SUDEP is because in addition to their epilepsy, they also die prematurely beginning around 2 to 3 weeks old, and this correlates with when their seizures begin. And so by the age of 10 weeks, you only have about 1/4 of the animals that are still alive. And so when I began working with these animals, I wanted to test the hypothesis that perhaps the seizures in these animals were leading to some sort of lethal cardiac arrhythmias that could explain or potentially explain their death. So we developed a technique to look at EEG and ECG in these animals simultaneously while they're awake and freely moving. And when we did this, we found that the knockout animals actually have a high incidence, a 5-fold increase incidence of cardiac conduction blocks during interictal periods. And so this is showing some examples of that where you can see that the QRS complex, which corresponds to the ventricle contracting, is missing 5-fold more often than in wild-type controls. And then we also tried to quantify this during seizure periods and found that the increase in these conduction blocks was even greater, although variable because not every seizure shows them. And so then we tried to get at whether these conduction blocks were intrinsic to the heart or whether they were being driven by the brain. And so to answer that question, we injected the mice with different autonomic blocking drugs such as atropine, which blocks the parasympathetic branch, or propranolol, which blocks the sympathetic branch. And when we did this, we found that the blocks would disappear with atropine, but not with propranolol. And so that indicated to us that likely they were being driven extrinsic to the heart, probably by the parasympathetic branch of the autonomic nervous system. And so as you probably know, the primary conduit between brain and heart in the parasympathetic branch is the vagus nerve. And so we went and looked for Kv1.1 expression in the vagus nerve and found that it's expressed in the vagal axons, especially in the myelinated fibers, as you can see in this immunohistochemistry image. And so then we went to try to record from these axons to see if the absence of this channel in the knockout mice predisposed them to having some sort of vagal hyperexcitability. And so that's exactly what we found. When we would give the, when we would record from single vagus axons and then apply just a very low dose of 4-aminopyridine, we would find that the axons would start spontaneously firing these kind of action potentials. They came in two types. You had the hot full amplitude action potentials that completely resembled a normal evoked action potential, and then you had smaller partial spikes that appeared to be triggered off of the full amplitude potentials. And so when we quantified this, we saw that about 75 to 80% of the knockout axons showed this kind of hyperexcitability phenotype, and only about 10% of the wild types. And so this showed that we know that the mice have hyperexcitable brains, and now we know that they also have hyperexcitable nerves. And so in one case, we actually were able to record a SUDEP event in one of our knockout mice. And so this is showing an example of a seizure leading up into this event. You can see that during the seizure, there's a very profound bradycardia and asystole, where the mouse's heart normally beats about 10 times a second, and here it starts where it's beating maybe once or twice a second. And then preceding the mouse's death, you can see where the EEG goes flat, and then the cardiac, the ECG gets very abnormal until eventually you go into cardiac arrest. And so this pattern of electrocerebral silence preceding cardiac arrest was interesting to us because in the one case, or one of the few cases where EEG was recorded in a SUDEP patient, this was the same pattern that was seen. So this is showing several leads from a patient who died of SUDEP. And so you can see over here the EEG goes flat, and although they weren't recording ECG, they could still see pulse artifacts, and so they know that their heart kept beating for at least 2 minutes after the EEG went flat. And so this is kind of the similar pattern that we've seen in our mice. And so that makes us think that perhaps the mechanisms could be similar. And so now let's talk quickly about the KCNQ1 gene, which is one of the, what I call one of the heart genes. And so this gene is kind of the inverse of the previous one we talked about. The previous one had a well-known role in the brain with high expression levels. Its expression in the heart was not really well understood. In this case, this gene had a well-known role in the heart and was not thought to be in brain. And so this gene encodes a KV-LQT1 or KV7.1 voltage-gated potassium channel. As I mentioned, it's highly expressed in the heart, especially in the myocytes and the conduction system. And it wasn't until we started looking in mouse and human samples that we were able to find that the gene's actually also expressed in the brain, and then this has just been overlooked. And so in the heart, this gene has a very well-defined role in mediating the cardiac IKS current, which is involved with repolarization. And so whenever this gene is disrupted in humans, the phenotype you typically get is called long QT syndrome. And so this is showing a normal ECG waveform pattern. And when this channel is disrupted, as you can see, this, the T wave here occurs later. And so it's prolonged. And so that's indicative of a prolonged repolarization. And that's one of the effects of this gene being mutated. And so in long QT syndrome, you have a very increased risk of ventricular tachyarrhythmias, which can lead to syncope, seizures, and sudden cardiac death. And so when we started looking at this, we were wondering if perhaps some of the seizures, which were thought to be cardiac mediated, might actually be mediated by lacking the channel in the brain, since we now knew that it was expressed there. And since we also knew that it was involved with sudden cardiac death, maybe some of these deaths could have been misdiagnosed as actually SUDEP. So when I was in Jeff Noble's lab at Baylor College of Medicine, we got in 2 different mouse knock-ins of human casein-Q1 mutations. And so the one I'm going to show here is one where you have a amino acid substitution in the S6 domain of the channel, which is the cause of human Romano-Ward syndrome, which is a type of long QT syndrome that's inherited in an autosomal dominant fashion and causes ventricular tachyarrhythmias. So when we started recording EEG and ECG in these animals, we found that the animals do in fact have epilepsy, which had never been associated with this gene. And so most of their seizures tend to be of the partial seizure variety, although they do have some infrequent convulsive seizures. And interestingly, when we looked at the correlation between EEG abnormalities and ECG, we found that it appeared that spiking in the EEG correlated with cardiac defects, such as right here, which indicated perhaps the brain could be driving these cardiac events. And I'd also like to point out that epilepsy hasn't actually been found or clearly demonstrated in humans with mutations in this gene yet. However, there's anecdotal evidence that shows that patients with these mutations do have, 14% of them do have some sort of personal history of seizures. They just may not have been EEG verified. And so, as with the other mouse, in the casein-Q mutant mice, we also were able to record a SUDEP event. And so, very similar to the other mouse, we saw that the mice tended to show normal ECG, and then so at some point, the EEG would become suppressed, and then you would see that the cardiac function would decrease until eventually you would hit some sort of electrocerebral silence followed by cardiac arrest. And so again, this was a similar pattern to what we've seen in that one human case, as well as in our other SUDEP mouse model. And so I'd just like to highlight that this group of long QT genes is actually very enticing when you think about genomic biomarkers, because there's at least 12 different genes that have been associated with long QT. We mentioned casein-Q1, but there's also 2 others in this list that have been implicated in SUDEP, casein-H2 and SCN5A. And I'd also like to point out that all of the genes on this list are co-expressed in brain and heart. And so I think that this could be a very good class of genes to look at with relation to SUDEP, since we already know all of them are expressed in brain and heart, and we already know that at least 3 of them have been linked to SUDEP anecdotally or in some other fashion. Also interestingly, as you can see, for those 3 SUDEP-associated genes, all of them are associated with some sort of seizure phenotype. And so this was a study where these are from patients who are referred to a long QT evaluation center. And so when they did that, they were quantifying how many of them had a seizure history or some sort of personal history of seizures. And as you can see, for those 3 genes, they all have some sort of positive seizure phenotype. However, it is important to note that with long QT, since you can get these ventricular tachyarrhythmias that lead to cerebral hypoperfusion, that can cause a type of cardiac-mediated seizure that's not actually epilepsy. So there's some complication in accurately diagnosing epilepsy in these patients. And so as we think about trying to identify genes that could be biomarkers for SUDEP, it sounds great in theory, but there's going to be a lot of hurdles to get over because of the genetic complexity and heterogeneity of epilepsy in general. And so part of the 2 things that I want to touch on is the fact that we all carry so-called disease mutations. We also have this idea of mutations that can be protective as opposed to harmful. And so I'd like to touch on both of those ideas. So this was a study that was done in Jeff Noble's lab a few years ago, where they sequenced all of the ion channel genes in epilepsy patients and then healthy controls. And they found that even in control people who are healthy and don't have epilepsy, that they carry several gene mutations that cause channels to have amino acid substitutions, even in channels that are linked to epilepsy. And so this is a histogram showing how many potentially harmful gene variants patients and controls have in genes that are known to cause epilepsy. And so that was kind of the surprising finding, is that even in someone who's healthy, for example, if you look at the number 7 over here, this individual actually has 7 potentially harmful mutations in genes known to cause epilepsy, and yet they're healthy. And so this confounds our ability to just simply decide, oh, you have this gene mutation, so you are going to have SUDEP or epilepsy. So it's because the problem is that we all, whether we know it or not, have gene mutations that could potentially, in a certain genetic context, cause epilepsy. And so the question here is, how do we decide what's harmful and what's beneficial? And then another thing I'd like to point out is that in the first mouse we looked at, which was the KCNA1 knockout mice, we know that there's several gene modifiers that can prevent SUDEP in these animals. One of them is a mutation in a PQ calcium channel gene, which actually, ironically enough, by itself causes spike wave epilepsy. But if you put these mutations into the KCNA1 mice, it causes a protective effect where they no longer die from SUDEP. Similarly, if you put knockout alleles for the microtubule binding protein tau into a KCNA1 mouse, it also has a protective effect on SUDEP in these animals. And so the question becomes, we may not just need to look at harmful gene variants for risk prediction, but we may also need to consider which gene variants could actually be protective and be indicators that this person may not be at risk. And so all of these add some sort of genetic complexity that we're just now trying to start to get a handle on. And so in summary, I've explained to you what SUDEP is, and I've tried to show you some of the physiological biomarkers that have been evaluated with this. And then we got into this idea of genomic biomarkers, looking at different candidate genes that could be used to try to predict SUDEP risk. And we talked about casein-A1 and casein-Q1, which are two voltage-gated potassium channel genes. And then as we just mentioned, even though this sounds really good, we're not quite there yet because, first of all of these genes need to be evaluated more strictly. And also there's a lot of genetic complexity that we're not quite understanding yet. But I'm hopeful that in the future, as we get more into the bioinformatics age, and as our ability to evaluate large data sets becomes more possible, that maybe we could start to understand some of this genetic complexity that's underlying this complex phenotype. Thank you.